This video is going to have a brief description of transcription for Gen Bio 1 at RCBC. I am going to speak generally, and later in another video, I will discuss the differences between how prokaryotes and how eukaryotes do this. Let's focus on the right-hand side. This is transcription here, which happens in the nucleus. That's the first part of the central dogma, that DNA is used to make RNA. In the future videos, I'll discuss how RNA is then used to make proteins. This process of transcription, as I mentioned, happens in the nucleus. We want to protect DNA from all the assaults that can happen out here in the cytoplasm. If we take a look at DNA, they're within a gene, within the transcribed regions of a gene. We have some specific sequences called exons and introns. That too will be discussed in a future video. What I want us to look at here is that we have a promoter region, and that promoter region helps to draw in RNA polymerase. We have some other regulatory sequences that can help RNA polymerase to find the promoter. Those sequences can be here, or they can be somewhere far away from the promoter region. At the end of a gene, the other end of a gene, we have the terminator region, and that is a signal that transcription should end. After transcription occurs, we then have a single strand of mRNA that is 5' on one side and 3' on another. Let's take a look at the DNA more specifically so that you can see here that, of course, it's a double strand, that the strands run anti-parallel to one another. So this 5' to 3' strand is sitting sort of on top, in this case, of the 3' to 5' strand. This is really important because I'm only going to use the bottom strand, which is called the template strand, to create my mRNA, which is here in yellow. And so what you'll see is that when these two strands pull apart, the bases within the DNA will be read by RNA polymerase, and the complementary base will come in and begin creating the RNA transcript, mRNA. And you can see that if we have a T here, the complement would be A. When we have A in the DNA, we bring in a U for the RNA. And C brings in G's, G's bring in C's. The reason that we only use the bottom template strand here is because RNA polymerase is like DNA polymerase in that it can only build the molecule of mRNA in a 5' to 3' direction. What that means is if I wanted to continue this strand, I could not add to the 5' end. I can only add to the 3' end. So new nucleotides are added on this end. That's what we refer to when we say these polymerases can only move from a 5' to a 3' direction. Let's go on to look at the 3 different steps. This here is initiation. Initiation involves finding the promoter, and the RNA polymerase is what needs to find that promoter. The promoter regions are somewhat similar in both eukaryotes and prokaryotes, and you may hear of specific sequences and something called a Tata box. That's a sequence that has a lot of T's and A's, and in particular, that's where RNA polymerase is drawn. Once it finds the promoter, you can see that the DNA begins to open up. And we are going to, as I said, just use one strand of the DNA. The other one's just going to sit there idle in this case. So once we've initiated and found the promoter, opened up the DNA, Now we can start adding nucleotides. And down here, it shows those nucleotides being added. We can look at it up here. The DNA is shown in red. The RNA is shown in yellow. Again, the new nucleotides are coming in and they're being added to the 3' end. Something that we may have not mentioned before is that in order for this to happen, you need what's called a free 3' hydroxyl group. That hydroxyl group is on the sugar of the 3', what we call the 3' end. And so that is why these new nucleotides can only be added here, because the 5' end doesn't have a free 3' hydroxyl. That is sometimes difficult to say. Okay, so we just keep adding these new nucleotides. We keep building a longer and longer strand of mRNA. And then we get to termination. And in termination, there are sequences within this region of the DNA. And what they do is they create this end of the RNA that folds back on itself. There's some complementary base pairing right here. That's called hairpin formation. And when that happens, it's a signal, hey, we've reached the end of the gene. The RNA polymerase falls off, the mRNA transcript falls off, and then the DNA can wind itself back up again. That is a general explanation of transcription.